introduction, thank you very much also for the invitation. Thank you everyone for being here for this presentation today. I think this is a subject that we all think is important for astrobiology, but it is also a very contentious topic. So hopefully I'll try to make sense of it and give you some ideas on how we can make progress on this huge problem of finding biosignatures of the oldest fossils. And I'll give you the example specifically of the Nuvvuagittuq jasper-carbonate banded iron formation. So let's see here. Why are the oldest traces of life on Earth so important? I think that there are biological reasons, like the first two. They give us information on the earliest evolution of microorganisms, as well as on the environmental conditions of primitive microbial ecosystem, but also geological reasons that inform us potentially on the conditions needed for primitive life to emerge on a planetary surface. And then for exobiology, astrobiology, this is very important for constituting a database of analogs for primitive extraterrestrial life, as well as ultimately, when we are pushing back the clock of the oldest fossil, it decreases the amount of time that's needed for life to emerge on a planet, because we don't know that amount of time. And therefore, it can potentially indicate how long it took on Earth and how many habitable environments and microbial biospheres that may be widespread and distributed out there, including within our own solar system. So pushing back the clock may mean increasing the probability that extraterrestrial life exists. So the major challenge is that we have to surmount the controversies that have affected the field in the past. There are vigorous disagreements over what constitutes biosignatures, and the answers you will get are usually different depending on the expertise of the scientist who talks about them. Then you have microscopic abiotic biomorphs that are very difficult to recognize from real bona fide microfossils. And also prebiotic chemistry is not very well understood. So chemical gardens are not usually recognized in the processes associated with prebiotic chemistry. So maybe there's a part of the theories for the origin of life in prebiotic chemistry that require being revisited, because at some point it does make sense that there would be some kind of chemical garden forming. That's a non-biological growth of self-assembled mineral structures. And this kind of is underpinned by this controversy that took place in 2002. In the same issue of Nature magazine, you had two different groups, one led by Martin Brazier, the late Martin Brazier, and by William Schopf, on the same microfossils from apex formation, so 3.45 billion years ago. And they showed very similar data sets, and yet they had the non-biological interpretation. That was the team led by Brazier. And there was a biological interpretation, which was favored by the team led by Bill Schopf. So this controversy has kind of dwindled, and over the years, a significant amount of new data has come about. But it appears that sometimes there are some that still doubt that there is any sign of life in the apex formation. So without going too much into the controversy, I think that the bottom line is that we need a multidisciplinary approach with multiple independent lines of evidence in order to distinguish microfossils from dubiofossils, those that we're not sure about, and pseudofossils, those that look like fossils, but that are probably really abiotic in origin. So that's a very important aspect. So how to do this? Well, we need to look at a range of characteristics. So for the Nuvvuagittuq Supracrustal Belt, we need to consider its the field relationships of the rocks that have been collected and studied and reported to have these microfossils. So we are located right here in the middle of this large pink-colored piece of Archean rocks called a superior craton. So this is in northeast Canada. So it is the largest part of Archean cratonic rocks. And in this, there's an astrobleme, which has never been confirmed to be an impact crater, but this Nastapoka arc here, just on the side of it, on the northern edge, there is this supracrustal belt which contains a linear belt of bedded iron formation that expands for about 3 kilometers distance. And this is embedded in this green colored rock. So the BIF is orange. The green colored rocks is this so-called Ujuaraluk unit, or it's been given other names like a faux amphibolite, but it is a type of amphibolite rock which has variable amounts of garnet, but which also has an extraordinary date associated with it, which is 4.28 billion years. Now, this is a date coming from samarium 146, neodymium 142 age estimate. So it's a model age, and it is not really the holy grail of geochronology in the Precambrian. The holy grail where we're thinking that we have a lot more precise and accurate estimates of ages comes from uranium lead isotopes in zircons. And these are typically in gneisses that are orthogneisses and like the tonalite, trondhjemite, granodiorite type rocks. And those ones, they can cut across the belt. And when they do, all of them that have been found are between 3.75 and 3.77 billion years. So this has been taken as the maximum age of the belt by some, but in some cases, it can be the minimum age because it is a cross-cutting relationship. And this is what Cates and Moyes say, but they don't think that this age is a real age of these amphibolite rocks. They think it's an inherited age from the mantle. So there's a controversy with respect to the age. We know it's really, really old. There are gabbros within the belt that intrude the belt that have an ages around 4.0 billion years. And there's several events of metamorphism that have been preserved in zircon overgrowths, as well as in resetted apatite ages. So they vary between 3.71 to 3.82 for the oldest metamorphic event, and then ages of 3.51, 3.35, 2.7 or thereabout, and 2.5 billion years for the belt. So these are probably events where you had either prograde or retrograde metamorphism. Prograde metamorphism would probably have taken place very early, because we do see quite a bit of evidence petrographically of retrograde phases and retrograde mineral growths, like like clinochlor, for instance, in the banded iron formations. So this is what they look like. The banded iron formations are quite extraordinary in the field. This is just showing a little bit of the diversity. Most of the BIF in the belt is actually dominated by quartz, magnetite, and amphiboles. So grunerite coming tonight. These BIF here are dominated by carbonate. These are the top ones. So they are orange-colored carbonates with chert nodules, like you can see here, some red coloring, as well black and some white coloring also. This is all chert. So microcrystalline quartz, ideal material to preserve microfossils, and embedded in this ferruginous, iron-rich carbonate from the Nuvvuagittuq Belt. And in some places, we see some of these nice outcrops of jasper that are embedded between, here in this case, two bands of felsic volcanic rocks. And we see a lot of white patches. So that's the remobilization of calcite, which took place probably during retrograde metamorphism. And here you see a contact between the two units. So with the jasper at the top, conformably overlying the orange-colored ferruginous carbonate BIF at the bottom of this picture. So this kind of association is quite common, actually, to see volcanic rocks with these kinds of banded iron formations. In the Nuvvuagittuq Belt, we have those jaspers. This is another picture of the jasper showing the fine laminations in the jasper. So these are strata-bound rocks. They're along strike of the main banded iron formation unit, and they co-occur in the vicinity of amphibolites that have deformed pillow structures. As you can see here, these kind of flat, rounded tongues of rocks, which are very similar to pillows that we see in the modern environment, for instance, in the Loihi Seamount of Hawaii, but also in the Lokenophiolite and many other places. Excuse me. many other places around the world where you have these kinds of rounded pillow structures in basaltic rocks. And here again, you have association with jasper, which again shows flat laminations, as well as in the modern, you have these finely laminated mats dominated by microorganisms made of siliceous and ferruginous material. So it's like a gel-like substance which hosts diverse microbial communities, including iron oxidizing bacteria like we see here on the seafloor. So this is a good model for us to study further. So we will talk more about these kind of uniformitarian comparison in a moment. But this is to show you the picture of the outcrop, what it looked like. It looked like a big bullseye in the field where you have a big patch of red jasper, and you can see the banding here going above the central red patch. So it's deflecting the sedimentary lamination above and below, typical of concretionary growth. So concretionary growth are understood to be diagenetic in origin, and in this case it would be diagenetic but still preserved through metamorphism, which is not that extraordinary because there are nodules and concretions in granulite facies rocks from other Eoarchean to Hadean terrains like the Saglec gneiss complex and Issoua. But this is extraordinary because it occurs in a jasper. So this was the sample that led to two papers that were made quite an impact in the media. First paper, so I'm going to talk about data coming from the two papers. The first paper in Nature was first authored by my ex-PhD student, Matthew Dodd. And this one came out just last year in Science Advances, where we describe the community and lay out a series of arguments to base our interpretation that these microfossils they contain are biological in origin. So now I should give a bit of context, geochemical context, to say that these BIF are nothing extraordinary. So starting on the left-hand side with those panels here first. At the top, this is a panel showing the trace metal composition of these banded iron formations compared between each other. So the NSB iron silicate BIF, which are typically more green and less red, and the NSB jasper carbonate BIF, which is in the red color here. The green one is from the Dales Gorge. So you can see these typical Western Australian BIFs have very similar transition metal profile for the relative abundances. So this looks like a regular banded iron formation, and same thing for rare earth elements. And you can see this panel here again showing the iron silicate BIF in green and jasper carbonate BIF in red. And this was a previously published data set in pink in the background. So reproducible data, data showing a positive europium anomaly with a positive yttrium anomaly, and not much of a cerium anomaly, not negative, not positive. Quite typical indeed of Archean hydrothermal fluids and hydrothermally influenced rocks like those in the Pilbara Craton of Western Australia, the Issoua Supracrustal Belt of Greenland, and the Churchill Province of Canada. You can see very similar trends. So when you see these kind of peaks, they are indicative of the similar kinds of processes that distributed the elements in this way. And lastly, for the sulfur isotopes, so the sulfur isotopes of sulfides in these banded iron formation shows a wide range of values. You have some banded iron formations with sulfides down to -5 per mil, and the jasper carbonate BIF, which are shown in red dots here, show a large range, including very positive values at +15 per mil or so. That kind of enrichment in 34S and range, large range in 34S, has been before ascribed to sulfur disproportionation, whereby the microorganism will take elemental sulfur and disproportionate it in a reduced and oxidized product. And that redistributes the isotope. It leads to isotopic enrichments in the sulfide that's produced, either from the subsequent reduction of the sulfate or the sulfide itself. But this range is not really consistent with magmatism or with abiotic processes that are typically invoked to explain much narrower range of isotopic composition. So this already indicates potentially that there's biosignatures in the sulfur cycle, but also that there was very little oxygen. So all the data points of this is small delta 33s versus small delta 34s. So if we look at the 2-dimension, if the data were to fall between those two lines, this would be considered to be mass independently fractionated. So here we have a lot of data points that are mass independently fractionated, and this indicates a maximum oxygen concentration in the atmosphere of about 10 to the -5, 10 exposed to the -5 present atmospheric level, so 0.0002%. So that still allows for some oxygen to exist, in my opinion. And that may become important later, but just keep in mind that oxygen levels were very, very, very low, if at all. But I do think there was a little bit. So yeah, well, finally, I should say now for the carbon isotope. So we just have done a bulk carbon isotope analysis for the organic matter that varies between -18, -24 per mil. And typical of banded iron formation, delta C13 of carbonate varies between -4 and -16 per mil. So we have indications that these BIF are typical of other BIFs. They have very low total organic carbon content. And these negative values are consistent with diagenetically oxidized biomass, because biomass typically has the negative delta C13 value. So having the carbonate also negative like this suggests in this BIF and the NSB similar conditions as these other BIFs I've been mentioning about, where we see similar signatures. So this is what the rocks look like here. These are slabs of different thicknesses that show the nodule that was specifically cut here. You can see the location of the targets. They're all inside those nodules. There's a large arborescent structure here that I will show in a few slides, which was fully located inside the nodule. And inside the nodule, in some of the cuts, we have these outsized blades of calcite. And you can see this here. It's color-coded in green in the Raman hyperspectral image, where the purple is hematite and the blue is quartz. And inside the calcite, we find micron-sized inclusions of apatite. Apatite with carbonate is an important mineralogical biosignature, but doesn't necessarily have to be. It's a possible biosignature. And we also find some organic carbon in the form of graphitic carbons in this calcite. So we'll talk more about this in a few slides. So these are some of the sulfides we looked at, with chalcopyrite that has these enrichments, and some of them contain apatite inclusions and galena inclusions, with some including with selenium enrichments. So there's some possibilities for looking at other things in the future, maybe selenium multiple sulfurides or multiple selenium isotopes rather. So in terms of the micropaleontological signals that we have in this rock, there are tubes that come with and without filaments and the jaspers from the Nuvvuagittuq. Here you have a picture. I don't know what's happening. Why is it so pixelated? But you see a parallel alignment of the tubes and a systematic diameter between the tubes. These ones don't appear to contain filaments, but these ones here at the bottom do contain filaments, and this one even contains a filament with some twists, which is typically what we see. These twisted filaments are not always in tubes, and tubes don't always have filaments, but they do sometimes, and they're composed of the same material. The wall of the tube is nanoscopic sized hematite, and the tube and the filament itself is composed of hematite with a bit of ferrous iron in it, and I will show the data later for this. Tubes and jasper is quite well known. This has been seen in the Ural jaspers of Russia by Maslenikov about 10 years ago. It's been reported also from the Loken in Norway by Gren and John Slack, and here reproduced by Matt Dodd in our paper as new observations as well, showing kind of undulated filaments of hematite inside tubes. Now, some of these filaments are twisted, as I mentioned, but the coil period or amplitude is not always the same. And you can see here a tightly coiled tube, and this is a combo transmitted and reflected light image. So you see in gray, the gray reflections is the hematite, and what's white is quartz, and the red is also the hematite, but where it's thinner. And this particular filament here going northward is also enclosed in a kind of a tube structure, although not very straight edged like we saw before. And in this case here in the lower left at a different focal plane, we also saw a number of kind of undulating periodic crystal alignment of the hematite. So here I drew a sketch over to kind of guide the eye that we have these kind of diagonally aligned crystals, crystal domains of hematite. And they're attached to a little ball of hematite. So this is usually referred to as terminal knobs, as it has been in the Milos iron formation in Greece and in the Figueroa jasper of the Jurassic of California. So there's a precedent for these kind of filaments to be attached to terminal knobs, although these ones usually are not twisted. Here we have twisted filaments attached to the knobs in the Nuvvuagittuq jasper. The twists come in a variety of shape and levels of preservation. Clearly, the rock, as I mentioned, has succumbed to several episodes of metamorphism. The maximum metamorphic event was probably at the lower amphibolite to upper green schist facies. And that has led to some recrystallization of the hematite, whereby initially these twisted filaments may have looked something like what you see here in the top right. Beautiful picture by Katrina Edwards, or late Katrina Edwards. And then these tubes here show an internal filament with a periodic arrangement of masses of hematite within. And here you can see also some more twists in the filament. So it's quite spectacular to see this, because really the crystallinity is distinct between the wall, which is very fine grain, and the tube and the filament, which is typically more coarse grain. So we have different amplitudes and periodicities of twisted filaments in the Nuvvuagittuq belt. Now, the twisted filaments co-occur with a wide range of other objects. So what is a dubiofossil? What is a pseudofossil? So at some point, we have to make a decision when we look at these pictures, and it's not always a trivial thing. But when we look at a lot of these pictures and many thin sections, we come to see things that repeat. And so these are selected among the best ones that I have taken. And you see here a twisted filament that co-occurs in a group of spheroids that have a central point and also an outer kind of corona. So they look like rosettes with circular concentricity. And even here, there's two of those that are fused together. So there is a circular twin between the two central oxidation spots, presumably. So we're not interpreting these objects as biological. We're interpreting them to be abiotic structures that form from chemically oscillating reactions. And I'll mention this in a little bit more detail shortly, but just remember here the twin. That's quite a striking feature. But they co-occur with the filaments, and they co-occur also with small groups of filaments that are parallel aligned. Sometimes they co-occur like here at the lower right. They occur with small irregular ellipsoids. So they're not really perfect ellipsoids, and they're quite large. Some of them are up to 20 microns and 40 microns sometimes. But they co-occur, the rosettes. You can see the rosette with the purple arrows in all of these images in association with other types of patterned objects in this rock. So one of the most extraordinary observation was that of a centimeter-sized structure of parallel aligned filaments that was branching up from a main stem. So here's my sketch of it, very primitive sketch of it, was showing the alignment, the parallel alignment of twisted filaments in red or straight filaments in blue. Those that were branching are branching up from the main stem, and then they seem to be pectinate branching. So pectinate branching on one side only. So that's what pectinate means. It means like a little bit like a comb. So all the filaments are pointing in the same direction, analogously to what's been reported as the bend stick structures of the Loihi volcano and from the Jan Mayen hydrothermal vent field in the northern Atlantic. But maybe the most similar ones are those from, again, from Loihi, but from a different paper than Edwards's paper, from Chan's paper, who looked at this long one here. You can see the scale bar is 16 millimeters. So these are centimeter-sized bundles of parallel line filaments of iron oxyhydroxides in the case of the modern environment. Here, in this case, they're all hematitic, but they extend over hundreds of microns, each and every bit of them. So, and they're organized in a similar way. And they are put at the same scale here. So this image with a 1 millimeter scale bar is put on the same scale as these ones. So you can see that there is a comparison that is relevant to be made here, certainly. And there's no abiotic mechanisms to explain this either, but I will come to this in a few slides as well. One of the things about filaments that we discovered is that the filaments are dominantly made of hematite, but when we look at Raman spectra of this hematite, we see a tiny peak here at around 670 reciprocal centimeters, which is indicative of ferrous oxide inside the hematite structure. So this is ferric-ferrous oxide, really, but it has the characteristic of hematite here with the peak that you see at 1,320 reciprocal centimeters, classical peak for hematite. We know this is hematite, but there are some subtle varieties of it, and you can see how it's mapped here, the yellow against the purple. And this has been actually tested on and experimented on by the group of Andreas Kapler at the University of Tübingen. And Aude Picard published a paper in 2014 that reported that when they take those twisted filaments and expose them to diagenetic temperatures and pressures, there will be some oxidation of co-occurring organic matter that leads to the reduction of some of the hematite and makes it ferrous. So we can see the increase of the peak here with these increasing conditions, the increase of the importance of that peak at 670 or so reciprocal centimeters. So another clue about the composition of those filaments is that they, so you can see here, this stems from a detailed work that we did with the focused ion beam, cutting slices of about 200 nanometers systematically and then reassembling all the backscattered images of the cross sections into a three-dimensional model. And the three-dimensional model here is showing hematite in blue. And what we interpret to be organic matter is this darkest material in the backscattered electron images. And this is color-coded in red in these images. So you have organic carbon or reduced carbon inside the hematite of these filaments. There's also a number of small quartz inclusion that are reminiscent of poikilotopic textures that are typically attributed to diagenetic processes in terrestrial sedimentary rocks. So another kind of object that's quite amazing, as if what I've shown you is not amazing enough. This is quite interesting because it's a different morphotype of fossil. Now we're talking about these irregular ellipsoid, not filaments, although they co-occur together. And in this case, the irregular ellipsoids, they form this group inside the granule that's located within the tree. So if I go back to this original image, this bit here is right in the middle of that parallel aligned bundle of filaments right here in the middle. So it contains hundreds of quite large-sized irregular ellipsoids. You can see a histogram of the distribution of their diameters here, basically showing that they average about 20 microns in diameter. That's the average of the long to the short axis of these ellipsoids. And you can see them in higher magnification here at the bottom. The cells are similarly composed to the tube, so they have kind of an outer wall composed of nanoscopic sized hematite, also sometimes with a bit of ferrous iron in it. And this is probably best explained as a dubiofossil because there are many abiotic biomorphs that can adopt shapes like this, irregular form, also clusters of this kind. So it's not trivial to distinguish them from real fossils since they have so few characteristics. However, they are similar in size to other known microbes that make iron oxides. This could include the Chromatium oceni, which are anoxygenic phototrophic bacteria that fix CO2, are involved in the nitrogen cycle. They're involved in iron oxidation, That's why they're often a little bit red, but they don't. And here in this case, you can see in the images they store intracellular amounts of sulfur in these little yellow spheres that are inside the cells here and there in these particular examples. So they're involved in all these biogeochemical cycles that are critical for the origin of life, and that may explain some of the data that I've been showing you. However, the rosettes, we've made a decision there. It was an interpretation based on this observation mostly, or observations of this kind, that they are abiotic, so that they are pseudofossils. So this is because they have a circular concentricity, very similar to what we see arising from chemically oscillating reactions. And they also have in cross-polars, it's best seen, this is the same place, just seen in cross-polar and transmitted light. In cross-polars, we see these radially aligned domains of quartz with these kind of gray colors, and it's all oriented towards the center. So this looks more like an orthogenic rosette that grew there in situ and abiotically during diagenesis, and that still preserved, same thing as the granules that you saw where the irregular ellipsoids were located, and the nodules themselves. So all these objects may all be abiotic. However, there's this increasingly developing model that we're pursuing in my group, which is to use chemically oscillating reactions as an analog for the growth of these kinds of objects, which I call diagenetic spheroids. Most people will agree they're diagenetic. If they're not biological, they must be diagenetic. And they're kind of spheroidal, but nobody really understands how they're formed, how these rosettes form, or the granules, how they form, or even nodules in concretion. There's still unexplained enigmas about them, such as the patterns they exhibit, for instance. So, okay, so we have microfossils, dubiofossils, pseudofossils. Are we right? Well, I don't think we can be wrong on all of them, but at least we have to make some progress. And part of making progress implies to test the null hypothesis. And that is to discount all the possible abiotic processes that are known before a biological interpretation can be reached. So this field has been growing from the work of Juan Manuel Garcia Ruiz in Granada, who has been experimenting since the 1980s and 90s with the growth of self-assembled mineralogical objects that are not the natural forms of minerals, but that are grown in the lab so as to know what is possible. And so the experiments have involved a lot of barium carbonate deposition or precipitation, including formation of twisted structures and segmented, elongated, filamentous, microfossil-like really structures, but they are compositionally distinct. In panels E and F here from Garcia Ruiz's paper in 2003, these are from the apex formation. So they're the controversial microfossils I alluded to at the beginning of the talk. And there has been several attempts to explain these forms using these abiotic biomorphs. So is it true? Does it explain everything? It doesn't explain everything. It doesn't explain everything because there's very low barium in these particular ones. We don't know where the barium went, and the replacement would have had to reduce the carbonate in the barium carbonate in the witherite mineral here in order to produce the carbonaceous material that composes these apex dubiofossils. For the tubes, there are explanations out there in the literature. Some work by Stone and Goldstein, 2004, has shown that bubbling gas through a solution that's supersaturated with respect to minerals that could precipitate out of it can lead to the precipitation of tubes composed of iron, ammonium, and sulfate. However, there again, we have a problem with the composition. We have some iron in what we're looking at, but ammonium and sulfate are not abundant in the Nuvvuagittuq jaspers. And also those tubes are not parallel aligned, and they do not show evidence for having filaments or with twists inside of their internally. Other experiments from Garcia Ruiz have shown, so these are from an older paper in OLEB in 1994, and this is from another paper in Science Advances. So they have done a significant effort to try to diversify the kind of abiotic biomorphs that have been that can be produced in experiments. So I've gone through all the papers, all the images, and here's a selection of the best images that could potentially explain or could potentially be compared to what we see in the Nuvvuagittuq Belt. But you see mostly radiating filaments. They're not really well organized. They're not twisted. Most of them are made of either barium carbonate, once again, or silica, like these little spheroids here. So these little spheroids look like they could potentially be similar to those that I showed as the irregular ellipsoid. And there's been some more recent work also from Rouillard and colleagues about growing these with pure silica, and they have been successful. And there's plenty of silica in the jasper, so that is a plausible growth mechanism for these irregular ellipsoids. So that's why we think the irregular ellipsoids might best be considered as dubiofossils with our current state of knowledge. But yeah, it's different from dendrites, from dendritic growth that form from diffusion limited aggregation, and also different from the range of more complex microfossil-like structures that were published a few years back in Science Advances, again by Garcia Ruiz. So overall, there's a lot of possible similarities, but none of the experiments creates all the different morphologies and patterns I've shown you in the Nuvvuagittuq. Those one from Julian Cartwright were grown in space in microgravity, again showing segmented features, all from solutions of metals, salts, which might be plausible, but there's probably not enough of the manganese, cobalt, and nickel to grow these in a similar fashion. Laura Barge looked at a few years back at growth of phosphate-based tubes, again through a bubbling mechanism, as Stone and Goldstein did. Again, with mixed results showing a range of diameters, parallel alignment due to gravity, but not pectinate branching, as we see. There are some pectinate, some branching filaments, however, in tubes that have been reported in the abiotic biomorph literature. This one by McMahon just a few years back probably is the one that comes the closest. This is a kind of an excerpt of the data set showing growth of ferrihydrite filaments at variable pH from 12 to 7. So a wide range of conditions are possible, and including some of them that are inside tubes. But there was no twisted filaments of ferrihydrite inside tubes of ferrihydrite that were able to self-organize. But some look like terminal knobs with filaments sticking out of them. So there is some good basis for comparing to these kinds of objects still. But I don't think that they are abiotic. I don't think that they are explained by any of those, mostly on the basis of chemical composition arguments and arguments based on the bulk of the morphology and the diversity of morphologies that we see. So these are examples of cabochon, of mocha stones, and different dendrites that we can see in museums. These are typically understood to be non-biological. Is it true? Is it not true? I don't know. But these, when dendrites are seen, they're typically interpreted by geologists as being abiotic. In my opinion, it remains an open question. But I don't think this is what we are looking at either in the Nuvvuagittuq Belt. So to characterize a little bit more the microfossil assemblages and the dubiofossil assemblages, we did a systematic Raman survey, and here's 5 different images of different, like the representative images of different microfossils and dubiofossils. In the top left, you have still the hematite in purple, the quartz in blue, and here the calcite is in green. And you have these numbers that relate to the numbers of the spectra shown on the right-hand side. And you can see that many of them, well, they all have the same characteristics, that they show a G band located at about 1600 reciprocal centimeters, and a D band for disordered carbon. So the G band is for graphitic carbon, and a D band at about 1345 reciprocal centimeters. And some of them look like graphite, like spectrum number 13 here. So we have true bona fide graphite in there, but we have also a number of occurrences of disordered carbons that have been relatively cooked. These kinds of signature with a broad and intense D and G band are quite characteristic of thermally matured kerogen. And this may actually be what we're seeing here because it co-occurs with the tubes in this case, with filaments and tubes here in this case. This is inside an irregular ellipsoid. You have an inclusion of different types of graphitic carbons that is quite disordered in this instance. But there is ordered carbon, and we have also several other examples of graphite in this particular rock. So what's going on? Well, we've been puzzled by some of the spectra we've been getting, especially in the branching tree structure. This is the same kind of field as I showed you earlier, with the main stem and all the parallel aligned branches that are branching off from it. And in fields in this tree, right next to the filaments, there are these little grains that are typically several hundred of nanometers in size. And if you look at how their spectra is exhibited, okay, so there's some spectra where you have organic carbon mixed with hematite, as in the case of 1, 2, and 3. But all the other spectra below are really unique. They have a strong and narrow peak at 1533, and two other strong and narrow peaks at 1457 and 1337. The peak at 1337 is that of crystalline sp3 hybridized carbon, which is from the diamond structure. But we don't think that these are typical diamonds because the diamonds typically don't have the peaks at 1457 and 1533. They just have the sharp and narrow peak here at about 1337. So what are these two peaks? We still don't fully understand what they are. We believe that they may be clusters of nanoscopic diamondoids. So that diamondoids are typically understood to be only one cage of the diamond structure, or two cages attached together, or 3 or 4, but it doesn't grow very large, because until you get to a certain size, you have a diamond. So I think that what we have is cluster groups of these small diamondoids that are known to form during thermal cracking of hydrocarbons, during higher grade metamorphism. So for instance, in thermally mature crude oil, diamondoids can be quite common and a useful indicator of the thermal maturity of the crude oil. So we think we find this here. And that's not so surprising in a way, because we have already crystalline sp2 hybridized carbon with graphite. Here we have crystalline sp3 hybridized carbon in the form of potentially nanoscopic diamondoids. But this was a new spectrum to me. I initially thought it was contamination. We did a very detailed analysis of all the components in the lab that we used to make the thin section. Nothing was diamondiferous, so it's not from any of the compounds we have. But it was puzzling. For many years, I've known of this, and I haven't published it because I did not know how to interpret it. And now we have found another occurrence. But first, I'm going to make some comparison between these new spectra, which are shown here in red and purple. And here at the top, you have a few cage structures showing the adamantane diamondoid and the two types of triamantane diamondoids. And you can see with the calculated adamantane spectrum or other calculated spectra from Oomans and Philic, there are some similarities in the sharpness and narrowness of the peaks, including the presence of the peak here for the sp3 hybridized carbon, the crystalline sp3 carbon, but none of them are identical. And that comes from his PhD thesis. Then we have all the spectra of nanoscopic diamonds and typical nano-diamonds and other kinds of diamond materials that were studied by Ferrari and Robertson. And there are some good matches with some of them, but the peaks in what has been reported so far are all broad. They're not as sharp and narrow as what we have. Indeed, what we have is the peaks are so sharp and narrow that one can consider them as peaks from a crystal structure. This needs to be a crystalline substance. the possibility that they are related to diamonds and diamondoids stems from that peak at 1337. The origin of these peaks still remains to be elucidated, but at this point we have early indication that these objects contain some nitrogen and sulfur, but we're still working on this with the nanosims. So to be continued. So then we have a little model to explain what we see in the Nuvvuagittuq Belt. So living biomass here, metabolizing, making biomass, making oxidized iron, as well as disproportionating sulfur. Eventually death and burial in siliceous gel, siliceous ferruginous gel. The banding may be attributable potentially to lysogang type diffusion, but I believe that the rounded objects, those diagenetic spheroids, granules, rosettes, and nodules, are indeed diagenetic and come from the oxidation of biomass, as in the case of chemically oscillating reactions. I think that's the simplest explanation to explain all the observable, the presence of apatite with carbonate, as well as graphitic carbons and so on. And then eventually during metamorphism, some of these crystals grew, the apatite grew, the crystals of calcite grew larger, but still preserved that texture and mineral inclusions that were associated with it. And this for the sulfides. The sulfides had inclusions of galena as well as apatite. So co-occurring in the same rock, but not inside the nodule itself. So for the diamondoids, we were very lucky to have this sample. This is my last data slide here. This sample came from Anshan in northeastern China. The rock is about 2.55 billion years, and it's been metamorphosed at the amphibolite facies. And as you can see in this section, there are bands that are outlined with this green-colored crayon type highlight, and these are bands that contain these parallel align and pectinate branching filaments of hematite and iron oxides here that you see. Other forms include these little bush or arborescence, which are probably best described as, or best interpreted as dubiofossils at this point in time. Here you have some tubes that are including branching tubes, but none of them contain filaments in this case. And you also have irregular ellipsoids, but in this case, they are larger in size. There are several, they're maybe 2 or 3 times larger than those from the Nuvvuagittuq rock. So there's some evidence for microfossils, dubiofossils, and potentially pseudofossils in this rock. But what makes it particularly interesting and relevant to what I was just saying about the diamondoids is that the same kind of occurrences are now seen in this second example. There's no other rocks on Earth, as far as I know, that has objects with this kind of spectrum. And you can see here the co-occurrence of calcite with the kinds of graphitic carbon and the same kind of spectra with peaks at 1533, 1455, and 1338 for also being nanoscopic diamondoids. So maybe a similar kind of peak metamorphism as well as similar rock type may lead to this. And this could potentially be a new type of biosignature to look at on other ancient planetary surfaces, crystalline sp3 carbon in nanoscopic size. So to summarize, I've presented to you a lot of different observations that suggest there's biosignatures and signs of life in this rock. Carbon isotopes for carbonate and graphite both support this. We have apatite crystals inside with graphite inside carbonates. We have rosettes, granules that contain apatite, carbonate, and graphite. Now, I did say that they were abiotic, those objects, but I think that they are still, that they require the presence of carboxylic acids in order to form. And that's because carboxylic acids are central in chemically oscillating reactions. They are the molecules that are the electron donors. So then there are microfossils that occur in Jasper concretions. There are microscopic tubes of nanoscopic hematite with and without filaments. There's filaments with variable twists and amplitudes. There are filaments attached to hematite knobs, pectinate branching filaments, centimeter-sized clusters of parallel aligned filaments, filaments associated with micron-sized carbonate, apatite, and graphite, There are these clusters of irregular ellipsoids. There's these enrichments in 34S in chalcopyrite, and we have uniformitarian observations. So ultimately, the conclusion is that they're quite complex microbial communities that seem to exist since the very beginning of the rock record, and they were capable of iron oxidation. So the main take-home message, or messages, is that solid cases for very ancient microfossils require dozens of lines of independent lines of evidence, as well as uniformitarianism. The new data is consistent with the ancestry of life dating back to at least 3.75, possibly 4.28, which is amazing. And then Earth's earliest life was a morphologically diverse microbial community that was able to oxidize iron, reduce sulfur, and living in hydrothermal vent environments. Now, I would think that the oldest microbial communities, therefore, these ones, increase the probability for the existence and the broad occurrence of extraterrestrial life. And that's it. That's what I had for you today.
Thanks, Dominic. Thanks very much.